Okay, let's get started. So thank you everyone for joining today. I'm Gabriela, as I mentioned before, and I'll be the host for today, while our main speaker will be Axel Martinelli. Martinelli, our head of biology. He will be the one walking you through the process of interactively analyzing and visualizing your omics data using our Omics Playground platform, which allows an interactive omics data discovery. Just before passing the word to Axel, I just wanted to briefly mention what we as BigOmics are trying to achieve. The main goal for the platform is to create a bridge in between biologists and bioinformaticians. Sorry for that. And with that being mainly to give a platform where both can kind of look into the data with biologists, being able to run the analysis in a faster way, interact with results, and plan next experiments accordingly, while bioinformaticians can allocate their time in a smarter way and hand over the analysis results to biologists in a more interactive manner. And that should foster a more efficient and interactive data discovery overall. So that's where Omics Playground was born, and that's how that's why we developed it firsthand. Our developers, they are bioinformaticians themselves, have been so since a long time. So the platform itself was born for that specific need into their original lab, and now it's available for scientists worldwide to use and expedite their analysis. As you will see today, the platform is designed to be very user-friendly and is specifically designed for transcriptomics and proteomics data at the moment, but we're expanding it to metabolomics support soon, as well as for multi-omics. Before we go on, the webinar itself will be lasting roughly 40 minutes, and we'll be leaving 10 minutes for Q&A at the end. Here are a few just quick links in case you need that and useful information. You can always register to the platform. There's a free trial account you can use to test the platform's functionalities and see if it matches your needs. And you can always reach out to us in case you have any questions. And of course, visit our website for any additional information. With that, I'll leave the stage to Axel. Axel, the stage is yours. You can start the presentation. Oh, just maybe one last mention. If you have any questions in the meantime and you just fear you'll forget them, you can also drop them in the comment section and I'll be reading them at the end of the webinar, or just when we will be having the Q&A session, you can just open your microphone and ask them live, whatever is more convenient to you. Axel, the stage is yours.
Okay. So hello everyone, and it's a pleasure to be giving this webinar to University in Finland. So the first thing I want to go through in the platform will be how to input the data. I will then give you a brief overview of the main visualization modules that we have in the platform. I won't be able to go through each one of them in detail as there are more than 150 figures and tables that we have in the platform. And so if you have any additional questions, we will leave 10 minutes at the end. So first of all, in terms of uploading and the types of data that we currently support, our platform is designed to be multi-omics. We currently support transcriptomics and proteomics data. We will soon be adding metabolomics data and a module dedicated to the integration of multi-omics data. So choosing the type of data is important here, as it will influence any of the steps downstream. So we will have analysis that is tailored for either transcriptomics or proteomics data at the moment. So choosing proteomics, then the next step is to choose your organism. And as you can see, besides human, mouse, and rat, we currently support more than 600 species. This includes all known or sequence vertebrates. We also have support for insects, plants, some protozoans. What is really currently missing is support for bacteria, something that we might address in the future. So I'm just going to pick an example data set, and the platform will require two main inputs from your end. The first input is a flat file that we call abundance table or read count table for transcriptomics data. In this case, I'm working with some proteomics data. And the structure is very simple. It's a file that can easily be read also with Excel. And it's normally produced by the units that will prepare your data, whether it's a mass spec machine or whether it's a sequencing machine for transcriptomics data. This is a pretty standard output. You will have your protein or gene IDs in the first column. And for human, mouse, and rat, we can support all different types of IDs, from HANCs to UniProt to ensemble IDs and so on. And then in each of the following columns, you will have your sample IDs with the corresponding abundances or read counts. Now here you can see that I have inputted normalized data, but of course you can also give us a raw data and we will take care of the normalization. The next step is adding the metadata that will contain the various conditions for your samples. So this is a file that you can easily prepare in Excel, and the structure is fairly simple. You will have your sample IDs, and all you have to make sure is that they match the sample IDs in the abundance file. And then in each of the following column, you will have conditions with their identifiers. So for example, here you can see I have activation state, I have time points, and I have a combination of the two. The next step is creating your pairwise comparisons. Now, for that, you can either upload a comparisons file, but unless you have particularly complex data, it's much easier to use our interactive interface. And this works very simply. You just choose a phenotype, or you can even intersect multiple phenotypes from your existing list. In this case, I'm combining activation and time. And then you simply move groups to the main group and to the control group. And you can, of course, also combine different groups. You don't restrict it to just one group. And then once you have decided which groups go into which of the two comparative groups, you can just edit the name and then add a new comparison to your list. Now, the next step is crucial because that's where you begin to distinguish between different omics data. And in this case, you can see that I have the protocol for proteomics data. You will have a similar view for transcriptomics data, but you will see that some of the steps will differ, particularly when it comes to normalization. So for proteomics data, we can impute missing values, which is currently done only using the SVD impute algorithm, but we plan to add more methods in the future. This is a step you can skip, as I know it's a bit of a controversial step among the proteomics community. Some people like imputation, some don't. So you have the option to remove it. For the normalization, when it comes to proteomics data, we use max median, max sum, and reference-based normalization. And max median is the default choice. For transcriptomics data, you will also see CPM and CPM plus quantile, with CPM plus quantile being the default choice. And of course, if you prefer to use your own normalization protocol, you can simply skip this step. The outlier removal is a more advanced feature. I would always suggest running your data sets with all the samples and then just checking that the statistical analysis is identifying what you also consider visually as a potential outliers. And finally, when it comes to batch effect correction, we have implemented 5 different methods. Now, some of you might know about batch effect and how to correct for it. So you will be aware of the fact that this is a bit of a double-edged sword in the sense that, yes, it can make your data cluster better, but it can also introduce false positives in your data. So I always recommend using caution when applying batch correction. And in our case, we have 5 different methods. We have two supervised approaches, combat and lemma, where you can define your batch effect. And then we have 3 unsupervised approaches, NPM, RUV, and SVA, where the batch effect correction is automatically done by the platform. So once you have selected all these options, the next step will be the one where you, of course, give a name and a description to your data set. And then where you can also select some advanced computation options. So in this case, you can see that the platform by default selects already a choice of gene tests and enrichment methods. Of course, you can customize this. For transcriptomics data, you will also see EDGE-R and DSIC2 among the gene tests. We also have some extra analysis that I normally run for all the data sets as it doesn't consume too much time. And we also have an advanced option that allows you to add your own custom gene sets or pathways as a GMT file, and they will be included in all the downstream analysis. So once you are happy with all your options, you can just click compute, and then the platform will take anything between 10 minutes to 1 hour, depending on the size of your data set, and will take you to the home page where you will see your data set appearing in the list of available data sets. And then all you have to do is highlight it and select load to load the data set into the platform. Maybe just quickly mention that it's possible with our platform to generate an automatic report as a PDF file in both a poster format or a slides format. And you can also customize this report by selecting the comparison summary and then selecting only specific pairwise comparisons. So this is an option that is available. And the other options that we also have is that you can share the object that contains all the visualization with another user by simply using their email address and then clicking on share. And even if they don't have an account on Omics Playground, they will be invited to create a trial account and they will be able to receive up to 4 data sets from you. Anyway, let's go to the visualization of the data. And as you can see, we have 6 different modules dedicated to this. I will go through the main modules used during the analysis. I'm not really going to talk too much about data view, but that's where you can find information about individual proteins or genes. But I will go straight to clustering because that's usually how you start the analysis of a new data set. And this is where you can visualize your heat maps, your PCA, TSNE, or UMAP plots, and we also included parallel plots, which are more useful for time series analysis. We currently don't support a full-on time series analysis, but it is something that will be available in the next iteration of the platform. So quickly, just to show you a bit the structure of our different modules, you will always see a settings sidebar on the right-hand side that includes options that will be applicable to all the plots that you see. In this case, we can choose which phenotypes to include in our analysis. We can filter the samples, and under the advanced options, we can also change the layout of our dimensionality reduction plot. So in this case, you can see that I have a UMAP plot, but I can easily switch between TSNE and PCA by just clicking the correct option. Now you can see I switched to a PCA plot, and as it happens here, we have a nice clustering between glioblastoma and healthy samples. Now there are 4 buttons that you will see at the top of your plots. These are buttons that allow you, first of all, to choose further options for customizing your plots. So here you can change, for example, the color and the label that you use for your plot. You can also add a second plot by changing the shape. So for example, I can add body mass index here, and now you will see I have different shapes available. And I can also move the label. So in this case, I might want a legend. I can just move the label to the bottom, and then I'll get a legend that describes the different combinations of colors and shapes. You can also turn this into a 3D plot. This is an interactive plot. You can move around and zoom in and out as needed. And you will also notice, so let me go back to 2D, that we have an inbuilt editor. Now this iteration is still a bit complex. It's based on ggplot, but in the next version of the platform, we will have a much more simplified editor that will allow you to do basic editing, such as changing the colors or the shapes and so on. So now, once you're happy with the visualization of your plot, you can always download them via the download icon as either PNG files, as PDF files that contain a description of the settings that you use to generate the plot, so that you can easily recreate a plot. And for more advanced users, you can also download the raw data that went into the plot as a CSV or Excel file that you can then work with in your own pipelines. And finally, I just want to mention that we also have some information about the plot, and this doesn't just provide you a description of what the plot represents, but also a detailed description of the methods that were used to generate the plot and the references for said methods that you can easily copy and paste in your document. So that saves you time from having to look up or ask for references when you write a paper. So quickly, when it comes to the heat map, again, we have, as you can see, a lot of options. In this case, we can switch between a dynamic interactive layout, or we can go to a more traditional static visualization of the heat map. Now you can see this is a rather dense heat map. This comes from a clinical study. We have a lot of different phenotypes. So we could simplify this a bit by removing the legend, but it still leaves you with a rather difficult heat map to read. So there are ways in which you can simplify that. Of course, we could remove some of the phenotypes. My favorite approach here is to split the heat map by phenotypes. So if I click this button, it's very easy to just focus on a given phenotype. In this case, the platform is going for the first in alphabetical order, but I want something a bit more meaningful, so I'm going to go for disease. And here you can see that in a matter of seconds, the platform will redraw the plot and divide it between, in this case, glioblastoma and healthy samples. And now you can see there is a much clearer structure to the data. Particularly, you can see that the healthy samples are very homogeneous, whereas the glioblastoma samples are a bit all over the place when it comes to protein expression profiles. Now, there are a lot of different options that you can choose here. You can change the mode in which we choose the proteins for the heat map. So you can choose standard deviations, you can choose principal component analysis. You can also select specific proteins or even add your own list of proteins or genes to generate a heat map. Now, where the platform comes into its own is when it comes to annotating your data set. And this is normally done by accessing various collections of gene sets or pathways. You might be familiar with some of them, such as the Go terms or the KEGG pathways, Hallmark collection. So in our platform, we have actually included more than 30 different public databases for annotation. So you have some popular ones like Hallmark or Go, but we also have some more specific ones such as aging or brain-specific ones. Here you can also see I have a custom database which I uploaded with my data set so that you can also customize this selection. So in total, we have more than 50,000 gene sets and pathways that you can use for analyzing your data. So in this case, I'm using the Hallmark collection by default, and you can see that the platform has split my proteins into 4 different modules based on their expression profiles. You can see from S1 to S4, and here we can then see the corresponding annotation of the 4 different modules. Now, if I change this, for example, to my custom gene set collection, you can easily switch, and you can see now that we have different names, and in particular for S1, you can start seeing some very interesting features that one would normally associate with a neuronal function. Now, of course, you could go through each one of them individually and play around with the different gene set collections, but we have introduced a new module called Feature, where you can perform clustering at either the gene or protein level or at the gene set level. Now, some of you might be familiar with this representation. In this case, I'm just gonna show it to you at the gene set level because this is also more biologically informative. And let me remove the labels. And so basically what we do here is rather than doing clustering by samples, we do clustering by gene set. So what does it mean? It means that we look at the expression for each of the given gene sets, and you remember we have more than 50,000 in our platform, excluding the custom one that you might have added on top of that. And we look at their expression across all the samples and then put them together if they follow a similar pattern. Now in this plot, the crucial factor is the colors. Red areas indicates hotspots of variation. So it means that you see a lot of differences in expression between samples. And this is really where you want to focus your attention. So now that you have that information, you want to look at the expression of the different modules across all your available phenotypes and see if you can identify a correlation between a given phenotype or condition and your hotspot of variation. So in this case, I'm going to focus on this tail here. So I'm going to choose disease outcome. And now if you look at the protein set signature, I'm going to magnify this. You can see that we have a clear difference in expression between samples or rather patients who have undergone remission versus patients who have died or have a persistent form of glioblastoma. So there is a recovery of function here and a clear loss of function here. So what we can do is we can go back to our protein set UMAP and highlight with a box just the very tip of this tail. And now in the table below, the platform will highlight only those gene sets and pathways that are contained within the tip of this tail. And here you can see now we get an overview of all the 30 different databases and the various gene sets that are contained within them. And as we go down the list, you can clearly notice various neuron function associated gene sets such as GABA synthesis. And this is nice that you see that in two different databases. So the overlap also gives you greater confidence in this analysis. You can see nicotine addiction from my custom database. You can see serotonin neurotransmitter and so on. So here you already purely visually get a nice overview of what's going on with your data set. And this is a feature that I like to show because it's unique to our platform. Now, when it comes to the bread and butter of analysis, of course, we do your standard volcano and MA plots. You can also customize those by changing the FDR and log4 change values. For example, we can choose some more statistically meaningful values. And you will see that the plots will be adapted accordingly. If you highlight a specific protein, you will see it in isolation in your volcano and MA plot, and you will also see some bar charts that will show you the expression in the pairwise comparison that I selected, as well as across all the other pairwise comparisons. In the protein sets, we'll also see all the gene sets that contain this particular gene or protein. And if you highlight one of them, you will then be able also to see where all the other components of this particular gene set or pathway appear in the volcano and MA plot. Now, one little thing I like to mention here is the star system. So you can see, for example, that this lymphocyte antigen 6 protein has 3 stars. And what does that mean? Well, if you go to the options, you will see that when I run this data set, I didn't just run it with a single statistical method, I ran it with 3 of them. And when you see 3 stars, it means that all 3 different methods identified this protein as significantly downregulated. So by intersecting the results, you get greater confidence in the robustness of your analysis. Obviously, this is a very good data set, but if your data set is a bit noisier and you don't get enough or any proteins or genes showing up as differentially expressed, that's not a problem. You can always customize that by removing 1 or 2 of the tests and then just relying on the analysis based on one of them, in this case, trend lemma. And you will see that the platform will immediately redraw all the plots according to my new settings. We also have a module dedicated to biomarker discovery. So this you can use to identify potential biomarker based on protein or gene expression using different combinations of machine learning algorithms. And I'm going to show you a very simple example, but of course, this is more interested for conditions where you have multiple groups in your analysis rather than just 2. So what the platform does here is produce 4 main plots. It produces a decision tree that tells you that in this case, expression of pimentin can be used to distinguish glioblastoma from healthy samples. You get a series of box plots showing you the top 8 biomarkers according to the analysis. And finally, you get this plot here, which shows you the cumulative variable importance of the different biomarkers as determined by the 8 methods that we combine. And they include 6 machine learning algorithms as well as 2 other statistical tests, so that the maximum score you can get is 8, and you can then see, for example, Vimentin has a score of 6, which means that basically almost 6 out of 8 of the methods identified it as the most prominent biomarker in the analysis. Of course, we cover gene set enrichment and pathway analysis. As I showed you before, we have a large collection of gene sets, and we can display this analysis. So this is actually very similar to the gene expression analysis or differential expression analysis module. And you can see here that we will use gene set enrichment analysis plots to show you the most up and down regulated gene sets. So in this case, we have here an example of a down-regulated gene set. You classically see this skew to the right-hand side in the blue area of most of the ranked proteins. And you can also see that the green line, which is the running statistics, is negative and also skewed to the right. And if you look at the positive or upregulated gene set, you will see exactly a mirror image of what I showed you before. So everything is now skewed to the left and in the positive part of the plot. Again, here we have a star system, and again, we combine different statistical methods to produce this analysis. So you will see this is a bit of a recurring theme in our platform in that we don't rely on a single method, but we tend to combine multiple methods to increase the robustness of the analysis. Now, variation of gene set enrichment analysis is pathway analysis, which here we perform based on the wiki pathways and Reactome databases. So basically, this provides you a more granular view of what's going on in a particular pathway. So you just don't get an information telling you, oh, this is up or down regulated, but you can actually see where the individually up or down regulated proteins are in your pathway, so that you get a better idea of what particular steps in these pathways are being affected in your condition. This is an interactive plot. You can zoom in and out, and you can also get more information about a single protein or gene by clicking on it. The same applies for the Reactome analysis. We also have here some plots, a bit more stylized than the wiki pathways plot. And you will also notice that we produce these activation matrices that basically allow you to look at the expression of the top, up and down regulated pathways across all your pairwise comparisons. So that it is easy for you to compare them and see if there are any differences among the conditions or which ones elicit stronger or weaker responses. Now, one thing in which our platform excels is in comparative analysis. And you've probably done a lot of Venn diagrams or seen a lot of Venn diagrams when you analyze your data set, when you're trying to identify, say, a group of genes or proteins that might be shared between your different conditions. So what we did here with our platform was extend this concept beyond comparing within your data set to actually comparing across all the data sets that you uploaded into the platform. And this actually includes comparative analysis across proteomics and transcriptomics data sets. So it means that you can actually compare not just proteomics with proteomics and transcriptomics with transcriptomics data, but also proteomics with transcriptomics data. And we do this by just looking at the fold change space rather than absolute values. And as I mentioned, you will be able to do this comparative analysis across all the data sets that you uploaded into the platform, but also across a public database that we have included in our platform. So first of all, this is looking now at all the collection of data sets that I have in my account. And you can see obviously the first match is with the pairwise comparison itself. And as I scroll down, I can then start seeing other data sets showing up. So for example, here I have the corresponding transcriptomics data of the same study. And you can see that the platform indicates a strong correlation between the transcriptomics and proteomics data sets. So that's quite interesting to see. And as I mentioned, you can also go to the public database. This is a collection of more than 6,000 experiments from the GEO database. So we have more than 10,000 pairwise comparisons that you can query in exactly the same way. And here you can see, for example, that the platform is already showing me a similarity between my proteomics dataset and this study on brain disorders. And in fact, the third most correlated dataset, the correlation here is negative just because they are comparing controls versus disease, and I'm just doing the opposite. But you can see from the name that this is actually another glioblastoma study. And in fact, the correlation that you see here is very similar to the correlation I had between transcriptomics and proteomics data from the same samples. And then once you have identified a suitable data set, you can really dig much deeper by doing a one-to-one pairwise comparison. So in this case, I'm going to pick the corresponding transcriptomics data set. And I'm going to pick, again, glioblastoma versus healthy pairwise comparison to show you just how we do it. So here we just update it quickly. So, but the default view is to produce a UMAP plot based on the, in this case, proteomics data. And then what the platform will do is it will take the transcriptomic data and overlay it on top of the proteomics data. That's why the two shapes are identical. But now the crucial information is the colors. So blue indicates downregulated, red indicates upregulated. And if you see that there is a correlation in up and down regulation, you should see that the red and blue areas are maintained. So there is not a loss of structure. And you can see that this is clearly the case, that the blue and red are maintained. You can further verify that by looking at the fold change correlation. You can see that I have a pretty solid correlation between the proteomics and transcriptomics data. And then here you can see the top up and down regulated proteins. And then you can see that they follow a very similar pattern at the transcript level. And finally, you can look at your genes or proteins of interest under gene correlation. So you can select them from the list. You can type a name, or you can, sort them by score. And for example, in this case, I'm identifying a gene or a protein where the correlation breaks down. In this case, I have a negative correlation with this signaling protein, which I can further visualize in this bar chart. And here you can see that there is actually an inverse correlation between proteomics and transcriptomics data, where I have upregulation in the healthy samples at the proteome level, but downregulation at the transcript level. So the final plot I'm gonna talk about, and maybe if I have a few minutes, I will show you the structure of the data view, is the systems biology one. And this is where we do more advanced data. It is split into 4 subtabs. The first one I'm going to talk about is cell profiling. This is more useful when you're working with single cell data, but I sometimes use it for my bulk data to identify possible contamination. So what it does, it will identify the type of cell you're working with. And as you can see from the list here, we have different databases that we can use. A lot of them are immune related, but we also have cell line, cancer type, and tissue type. So if I select tissue type here, you can see that it tells me, well, it's brain tissue, but it matches foremost with liver, and brain is only down the list. But there is a good explanation for that. And we can visualize that more when we go under mapping. And again, if we select the same database, so here you can see that the strongest match is with liver, and then we have a strong match with brain. However, these matches are very much dependent on the type of conditions we are looking at. PT actually refers to the healthy samples, and as you see, for the healthy samples, it is correctly identifying brain as one of the top matches, whereas for the tumor samples, it looks like liver. And if we further explore it here with the abundance plot, okay, I'm going to choose disease, you can finally see that whereas the healthy samples are basically identified exclusively as brain, the glioblastoma samples are all over the place. And this you would expect, as these are cancer cells and it will start expressing markers that do not belong to the brain. The other component of this module is gene or protein network analysis. And we do this in two ways. We either use WGCNA, which some of you might be familiar with, and we have a lot of plots that are displayed here. The one that I normally use the most is the module plots, as it provides you with some biological insights in your data. So basically, the platform will use default values to produce various protein modules in this case. And then what you can see here in this heat map is a correlation of these various modules with any of the existing conditions. So in this case, for example, you can see that ME1 has a strong negative correlation with healthy samples and a positive correlation with glioblastoma samples, and ME2 is basically the reverse. So these are two of the modules I might want to look at. In this case, I already have ME1. And if I look at the functional notation, I can, for example, see that there is the upregulation of angiogenesis in glioblastoma, which is typical of tumors. You can also see cross signaling being affected, inflammatory responses, hypoxia. These are all typical responses that you will find in tumors. Another way in which you can do protein network analysis is by using the price collecting Steiner-Forest algorithm, which is based on the string protein-protein interaction network database. And here you can see that the platform has generated two networks based on the up and down regulated proteins. These are interactive plots, so you can highlight a specific protein, in this case vimentin, which, if you remember, was one of my most prominent biomarkers. You can also stretch it around just to facilitate the visualization. And the last module I'm gonna cover here is the drug connectivity map. Now, this is a more advanced type of analysis, and some of you who are not working with drugs might wonder, well, why should I even bother with this one? Well, actually, this is more flexible than you think. It's not just used to identify the mode of action of a potential drug you're working with. It can also be used to identify drugs that could be repurposed for treating your condition. We have also included not just the L1000 drug connectivity map database, but the L1000 gene perturbation database, as well as the CTRP and GDSC drug sensitivity databases. So even if you're not interested in drugs, you can use the L1000 gene perturbation database to identify genes whose up or down regulation might be driving the condition you're interested in. And just to quickly show you an example of the usage. So obviously in this case, I'm looking glioblastoma versus healthy. So I'm not interested in the positive correlation, as this will indicate drugs that basically can recreate a glioblastoma profile. But this is useful, of course, if you were working with a drug, in which case this putative drug would be related to cisplatin. Here I want to look at the negative correlation, so I want to look at drugs that could be used to treat glioblastoma. So for example, one of the top 3 most prominent drugs is entinostat. And you can see it has a strong negative correlation, so strong inhibitory potential. And we can then further simplify the analysis by combining all the drugs by their mode of action. So we perform a meta-analysis, and here you can identify the most positively and negatively correlated modes of actions. And in this case, you can see that histone deacetylase inhibition is the most inhibitory mechanism for glioblastoma. And this was actually also verified in published data where histone deacetylase inhibitors were used to treat patients. When you look at the gene perturbation map, you will see that it will provide you with some useful information about what's going on. So in this case, this gene perturbation map includes both overexpressing and knocking down gene expression. And here you can see that among the top most positively correlated genes, I have basically cytokines that have been upregulated. So for example, you can see the top one is interferon gamma. And this is not surprising considering that you have a lot of inflammatory responses going on in glioblastomas. But again, if you're working with different conditions, they could also highlight genes that could be, for example, used as a target for intervention. And finally, just briefly mentioned the drug sensitivity database. Now, this is definitely a bit more niche, but if you're working with a drug and you're interested in identifying suitable partner drugs that could have a good synergistic effect with your drug of interest, then you will want to use the drug sensitivity databases. As is in this case, the partner drugs are suggested by the positive normalized enrichment score. So basically, you will want to look at all the ones that have a positive correlation. Obviously, in this case, I'm not looking at the drug, so the plots are not great. But basically, if you were working with a drug, it would suggest that any of these could be good partner drugs as they would counteract the expression profiles that are elicited by the compound that you're using in your experiment. And I see we have only 10 minutes left, which is exactly the amount of time for the questions. So I will stop here and please feel free to ask any questions.
Just a reminder, you can also just write them in the chat if you don't feel like turning on the microphone.
No questions? I don't know if I overwhelm you with too much information or if maybe Finnish people are a bit shy.
Well, in both cases, so as you can see, today's session is recorded, so I'll be sharing the recording most probably tomorrow morning with you, so you can rewatch every bit of it if something was not, or if you missed some parts of it, you can just rewatch it, and most likely maybe questions will also come afterwards after you kind of digest the information. So feel free to reach out anytime if you have any doubts or questions about the usability of the platform. We're always here to support you. We have several contact forms on the website, but you can also reach out directly to us or just reply to the emails that we've been sending it. I see there was a question. There's a question actually last minute. I know, Kaisa, I hope I pronounced it correctly. The question is, how about analyzing the longitudinal or other autocorrelated data?
So one thing that I didn't show is that you can do some correlation analysis in our platform, and it's actually I sort of skipped that for time's sake, but it's actually something that I know some of your users are using to a great extent. So basically here you can select a specific protein or gene from your list. So maybe I'll just pick albumin because it's one that I like in this list. And you can see all the genes that have a strong, in this case, positive correlation in terms of expression with albumin. So here you have a list. And then you can also visualize that as a correlation graph that shows you both positively and negatively correlated genes. So based on the color of the connections, you will have red ones indicating positive correlation, blue ones indicating negative correlation, as well as a correlation UMAP that shows you, in this case, the list of the most positively correlated in this little island here and the most negatively correlated, obviously, in the downregulated genes here. So this is where you can perform one type of correlation analysis. There is actually more about that when you go to data view, which is the plot I didn't show. So again, here, if you select a specific gene, you can also see the top positively and negatively correlated proteins based on the data set. And there is also quite a lot more information you can get about an individual gene. You can also see the protein abundance. So for example, maybe let's do it by disease, or you could do it even at the individual level. And there is a lot of other options that you can customize here. But yeah, this is where you can see some correlation between the different protein or gene expression profiles, as well as, in this case, tissue expression.
I hope that answers your question, but feel free to add any follow-ups on that.
Yeah, if there are, if you need more details, you can always write to us and we can provide you with more information.
Okay, good. Then I would say, given that we are also a bit over time with the minutes, I leave it to you. If any questions come after this webinar, you're always free to contact us. We're always available to answer any of your doubts. I'll be also sharing all of the links where you can access the platform, all the relative tutorials and anything where you can access some of this information we shared today, but you'll always have the recording anyhow if you want to look back at something. And yeah, thank you so much for joining us today, and I wish you all a pleasant day and hope to hear from you soon.
Yeah, thank you all for
Thank you.
joining.
Have a good day.